the end, we built all this story during the pandemic, so it was only the Zoom things. Each people we never met in person, in fact, and just in June 2021, when we have the party to celebrate the paper, we can meet the people in person. So that's the that's the end of the story. Thank you.
Thank you very much for this very nice talk. Are there any questions? Okay, yeah. We have time for 2 questions.
Hello. Hi. This is a question from an experimentalist who collaborates with people from data science, but more like old-style data science. So a typical example is segmentation of surfaces. That's my case. And when they do that, they come up with very simple fitting, and they come with a very simple model, let's say a Gaussian model, and they have a noise, and then they can use a whole battery of tools from estimation theory, information theory, and to get an understanding of what's driving the accuracy of the estimation. And in a way, I think this understanding is more important than the result. And so my question is that with deep learning tools, do you have also this understanding of what's driving the accuracy, or do you just have a result of, let's say you estimate a surface? And I know there are tools using deep learning to segment surfaces. So it's really a naive question from someone who doesn't know the field.
No, the answer is okay. Until now, I should say. There is no really theory. So it's, as I say, it's really a black box that we inject data and we obtain a result that makes sense. Okay, and when, of course, we have the validation, so we check that the system, but there is no really explainability of what it's inside. But recently, and the kind of thing that I have tried to do to make to attack the system, to provoke the system, and this kind of things, we start to a little bit know a little bit better what is the limit. In fact, it's more a question of the range of the data that we can inject in the system to obtain a good result. So we more elaborate like that. But there is no really mathematical formalism or something.
All right, I see. Because it's sad because in a way having the understanding allows you to bias the experiments to go into the best direction.
Of course. Yeah.
Okay.
Yeah, right.
Okay.
Yeah.
Thanks.
Yeah.
All right. Thank you very much. I think we're time.
Yeah, thank you, Daniel. So I have a question on the attacks that you have shown. So some attacks actually did not do very much. Yeah. So there's so we have seen that for some attacks actually the results were still very good, and for other attacks they were not. And so my question is, did you use specifically in data augmentation intensity changes and Gaussian blurs, for instance? And is perhaps the robustness due to the kind of data augmentation you have used to train the model?
Yeah, true. I do not. Okay, it's just a preliminary stuff that I did 2 weeks ago. That's an interesting point that I do not think too much. But also the thing that we can also ask ourselves, so if we make an attack so that the system starts to crash at some point, so maybe we can also use this data to train again the systems and to see if we improve the robustness. So it's another way, but probably there is a link with data augmentation, true, yes, that's probably true. I do not have a specific complete analysis, but we can perform this kind of a study. I think and it's very important for our field also to test a little bit the range of the data that we can put, and of course the kind of degradation that we have on microscopes. So, yeah. Mm-hmm.
All right. Thank you very much. All right. me a mic. While our next speaker is getting ready, just going to introduce her briefly. So our next speaker is going to be Perrine Polgilotto, that is working at the Université de Nantes. She's a research engineer in computer vision and bioimage analysis, and she's working on multimodal image analysis. Let me get my notes because I forgot. And she's also heading a microscopy facility that is working for histology, photonics microscopy, and image analysis. And today her talk is entitled Multiscale and Multimodal Registration, an overview of methods. There you go. And you'll have 45 minutes, if it's okay. Yeah, go for it. That's fine? Okay. All right. Thank you, Perrine.
Oh. Then it's good. Okay, so thank you very much for the introduction. Then what we're going to talk about now, it's really to focus on one particular task of bioimage analysis, which is registration. Ah ouais, merci. I was going to die. So yes, focusing on one particular task of bioimage analysis, so registration, and we'll see, the purpose is to see what is image registration, what are, I would say, the classical or approaches for registration, where is deep learning coming in image registration, and also as Daniel was also underlying, how you do validate this kind of image registration, because here we are going to talk about multimodal registration and multiscale registration, which is basically some kind of hard task. So what it is, so image registration is can be defined by the computation of the best transformation allowing to align or superimpose two images and applying it. By images here I mean any images that you can have in a microscopy, so usually they are multidimensional. And something important to understand also is that it's some kind of different problem to do monomodal registration or multimodal registration. So monomodal means coming from the same microscope, for example. So just for you also to see that you all know or have used image registration, and to see also that it's well used in other fields. So for example, in medical field, image registration is something that you use when you do image fusion, like combining information, MRI or functional MRI. It's also something which is done like from years for what is called longitudinal studies, such as studying a patient over time, for example. And something very classical also in the medical image field is creating atlases, in particular for well-known organs, I would say, like morphological speaking, like brain, and to match, for example, a patient to this atlas. And I'm talking about medical image field because they are doing image registration since much more time that we started to do it. In biological image field, this is also something that you use, I guess, routinely. For example, if you do motion compensation, like you have some kind of drift, basically. That could happen, but also it can be also like you have several capture the same sample and it's deforming. That's also the kind of tools that is used for some kind of light sheet imaging when you need to reconstruct on from different angles the full volume. Also what you do when you do chromatic distortion correction, like correcting different channels, so usually using beads but not only. For people doing electron microscopy, it's used a lot also for any serial section reconstruction, and also coming to like microscopy, and everything related to stitching, mosaicing, This is all based on the same kind of approaches, which is image registration. And the one I'm going to focus on is the multimodal image fusion. And the more classical, I would say, flavor of it is a correlated light electron microscopy. But there is any microscopy and any modalities could be merged. So what are the interests? I guess all of you maybe have already heard about correlated multimodal imaging, but basically the principle is simple, the method is not, but the principle is simple, is that you're going to get the same specimen and imaging it with different modalities which are bringing very different information about the samples. And usually you try to merge structural, functional, temporal, chemical information. Also, you have the notion of having a lot of different resolution covered. And what you want also is to maybe have different contrast mechanism. It's used in particular, for example, when you have also aware events that you need to identify for further analysis or to identify specific region of interest. And just for you to know, there is mainly two approaches. So of course, there is some machine, I would say, which are combining different modalities. So there are some kind of pre-aligned, so there is not that much of registration to do, but the more not classical, but the more used and the more maybe powerful way is to do sample transfer, but this is some kind of very difficult and very pluridisciplinary. A lot of different steps have to be taken into account, and here then you transfer the sample between different modalities, and then you need to register all images. So just to give you an example before going into more details about registration. So I was saying there is different purpose for correlative microscopy. So here is one example of application where the purpose is to combine information you could get only from one modality. So here it was work with some people from Germany using a specific line on a synchrotron in the UK. And what they wanted to check was some virus absorption, some virus pathway into the cells, and basically they needed an indicator of the endosomal membrane disruption that they would not get or would not see from the soft X-ray imaging on the line. And so they do add some markers, and then they froze the sample at different time, and the purpose was then to find back the area image from the light microscopy image. I'm going fast here, but basically at the end what you get is some overlaid of the tomograph obtained, so it's all in 3D, so the tomograph obtained from the soft X-ray specific line of the synchrotron, and of the structured illumination microscopy that was done on the cryo-sampled in order to check which membrane are disrupted and everything. So I'm going a little bit faster. And this kind of study that allow merging these two modalities to, for example, study at different stage after infection, which vesicles are disrupted or not, the size of them, both by the resolution of the soft X-ray. And you can find some very, for example, new structures that you could not be sure that they contain virus if you did not have the virus staining as well and the disruption. So this is one example, bringing different information together. Another kind of application, and I will explain why I'm mentioning that. It's also that sometimes you want to validate or understand a new modality or a new biomarker or a new probe, and for this you're going to use a known one and compare with a new image. And this is an example of some modality which is based on OCT, but dynamic OCT, and the purpose is just to understand what we are seeing here. So this is the color code. The colors are coding frequencies actually of vibration, and the idea is just to see, okay, which we see something, are there nuclei, are there cells, which kind of cells, and this kind of thing. And for this you can use also correlative imaging in order to use a known staining, giving you some known cells, and trying to register in 3D just to check, okay, what are we seeing, which kind of cells and everything. And it can be kind of accurate. You can see the area which are not vibrating, for example, which are the more dense here also in the nuclei staining. And for this kind of approaches, and you will use, for example, the nuclei that you could identify for sure, and you know where are all the nuclei in the reference image, and then bring this segmented nuclei to the new modality, I would say, or new use of the modality to better understand what you're seeing by doing some intensity measurement, for example. So this is two kind of question you're asking, and there is different approaches. And what we have seen from the two examples is that the content can be very different, and also sometimes you don't want to assume that the contents are the same because you want to check if this is the position or not of what you are interested in. And there is mainly two approaches for registration. I would say one is intensity based, and the second one is what I would define later, fiducial based. But basically, fiducial would mean something that you extract from the images specifically, and that you are going to use for registration because you know how it looks in the other image. So you know it for sure. This is something you impose. And not fiducial means anything which is not used for registration. And this extraction of fiducial can be automatic or manual. And if it's some kind of known because you're pairing them, and some people are manually pairing fiducials, and you can just compute the transformation, which is not that simple, by the way, it's not a just. Or you can also kind of try to propose yourself the matching. When you go to any of these approaches, these are the different bricks of a registration, which purpose, once again, is to compute the best transformation linking the images, and we're going to see where is deep learning now in this kind of different bricks. So this is a team, I would say, which purpose is to find the best transformation. And for this, there is usually one image that will be modified, one image on which you want to transform this image on. And you need something which is called the model of transformation. We'll come back to that. And you have some kind of a team member which is actually going to say what would be the next proposal of transformation parameter to try, and who would say when it's the best one. And you have also something to define what is best. What does it mean, being the best transformation? So here, for example, this is called a metric, distance metric or similarity metric. And the most simple, for example, to think about if you're using some fiducials, that would be to check what is the minimum distance between the points after you apply the transformation, and then you go on until you find the minimal distance. In intensity-based registration, basically there is a lot of them. These are classical one, I would say, but are not the one the most efficient. So here, if you take two images, so one is from EM, one is from bright field, and here you can see you are lucky enough to have some very characteristic and visible stuff that appear in both images, so which were melanosome here. And just to give you an idea about what is a metric, so the purpose, if you are looking, for example, just for one parameter of the transformation, that would be the translation. Okay, you can see that when the metric evolves, for example, here one simple one would be just the difference in pixel. So for a monomodal case, that will works very nicely. So you find a very big minimum here. And if you go for the multimodal one, okay, you can see when they are supposed to match, nothing happen or nothing very characteristic. That will not be that obvious to find for a software then. There is some other metrics, and for example, correlation in that case would be kind of allowing you to get a maximum even in the multimodal case, but most of the time it's not that simple because you don't have that much of information. So we've just seen that monomodal is more easier to find a metric which is going to work very easily for it. So one approach where deep learning is helping a lot, for example, it's basically to try to get back to a monomodal problem. So basically what you say is that you have an EM image, for example, and you have here a structure elimination microscopy. So this is works from these people. I didn't contribute to this work. And you do use a lot of prepared image, meaning that you had a lot here. It was about 30 images of pre-processed, pre-registered image, registered image, and then you try to learn, as Daniel was showing you, what would be, you try to predict what should be the fluorescence image from this image, based on the learning that you did it. So you do that, you try to learn. So here you don't really care about artifacts or hallucination, because the purpose is really to get back to something that would be very similar, so you can use the classical approaches for monomodal registration. And then when it's done, you just apply classical methods and to any new images, try to predict what would be the signal here, and then apply it and compute the transformation and apply it as well. So here you play, and the transformation of the image itself to come back to a monomodal problem. There is also other approaches where basically you try to learn the metric by itself, because we've seen it's not that easy to find a metric. So there is some work. So here's from the medical field, and Diana also was is going to speak later in the day. But here, for example, you're not going to try to learn the registration or to predict an image. What you're going to predict, it's a dissimilarity map. That would be some kind of the matrix that you will be using and trying to then minimize. And this is the one that you will learn by deep learning. So that's another place to put deep learning in. One thing to sorry. One thing to know also, but I could not find any example, but some people are trying in some other field just to consider registration as a problem that could be solved by deep learning itself. So basically you give a lot of example of registered image, and that's it. And you try to learn the transformation directly. So just to come back also to these approaches for registration, we said there is intensity based, and here are other also examples. So with the same principle as the one we have seen with the chromatin, where you do train based on a pair of images in order to predict what would be the monomodal problem, and you apply the monomodal problem. To mention also the same kind of idea of going to a monomodal problem can also be achieved without having to use deep learning, and there have been interesting work, for example, from Toledo, Boutemi, and all, which are actually trying to find also a common representation space, but just based on, I would say, more statistical tool and probability tool, trying to basically, in a few words, it's some kind of a Laplacian of Gaussian, for people who know that. So it's just a Laplacian of Gaussian filters, but which different parameters are automatically estimated from the images, but without any deep learning here. So that's a different thing. Just before going to the next part, so there is a lot of different approaches. Not all of them are implemented as a usable software. In UBIAS also we have been doing a directory, I would say, or a database of software that are usable and that you can use for different tasks. So it's called be.eu, and you can look for any kind of task or any kind of modality for a software that could work for you, which is implementing some of the method I was talking about. One thing also to know is that, okay, we've seen how we could compute a transformation with or without deep learning, and then and that will ask then another question, which is how you validate what you're doing and how you estimate how accurate it is. But basically, the the question is, okay, what do you do with the transformations that you have done? So the most classical way of using the transformations that you have computed is basically to transform one of the image, okay, and then overlay them and maybe do measurement on this overlaid image. Or the only thing that to be aware of is that when you do such a thing, at some point you are going to invent some pixels because you're going to add pixels, and at some point you will take some decision about the pixels that you're adding, so which is called interpolation, which is not bad by itself, but just to be aware of. Another way also, and this is a thing which is mostly used when you want to look at your data, is basically not to transform any image, but just to transform the way you're looking at it, and this is preserving the full resolution of the data, just changing the way you're looking at it. And last way, and I think this is usually super useful because this is what you want to do with your data usually at the end, is you do not transform the images, you just transform a region of interest, and then you keep your original images, not touching any intensity, and you just transform the area where you want to do the measurement. So you will be sure to be exactly at the same area, and then do the measurement exactly at the same area. I just want to spend some time about a super important problem, which is how do you check it's good what you have done for registration. And this is not an easy problem, in particular in multimodal registration validation. So in multimodal registration, because when you do multimodal registration, there is no ground truth most of the time, or only approximation of it, because we are using microscopes, and so we have not an infinite resolution. We don't know exactly where are the object of interest, and this is recognized as a difficult problem, just to validate it and to check if it's good or not. At least to estimate how good we are. It was mentioned during the question also that it's important when you measure something to know how far you can be from the reality, or also maybe how many uncertainty you could include in your model if you do some measurement. And the difficulty also is that intensity metric scoring by itself is maybe not adapted. And why is that? It's because we are doing multimodal imaging because we want to bring together very different content, or we want to check if a content corresponds to another one. So it's very hard to take the assumption that they are the same and to use something which is going to check or correlate or measure the correlation to check the correlation itself, because you're using the assumption for validation otherwise. So it's not that easy, and also we know that you're going to have errors in your registration. This is for sure, and this is for several reasons. And one reason is because, of course, your sample may evolve between two modalities. It's also, as we say, very multiscale, and the consequence of that is that sometimes one pixel match 100 of pixels. So there is an uncertainty also due to this pixel size. And also because of the resolution of the microscopes themselves, there is no way you can locate perfectly a structure of interest. And then even when the sample did not deform at all, you will have an error. I'm just going to explain that. And this error, so as you know, when you're locating something, for example, in fluorescence microscopy, basically you have some kind of a Gaussian likelihood to find the good answer, I would say, and there is an error which can be represented as this Gaussian, meaning that, okay, you may find the good answer here, but you are also likely going to be somewhere here. And this is the resolution, basically, of your microscope, meaning that when you will localize something, either an intensity or any fiducial, meaning any points that maybe you would add to your images to help the registration, then you will never get exactly the good localization. And when you do that, even if the two images are perfectly matching, the the consequence of that is that you will get an error in the registration. Okay, even if the results were perfectly matching, you will never get the perfect answer. We will see that also in a regression. So how do people either I would say biologists or people doing computer vision check the accuracy, like on real data? It's not that easy. So usually what you try to do is basically keeping some objects where you know where they are, okay, and you will check on these points not used for the registration what are the accuracy. So you keep some of them apart, and you're going to measure the accuracy on it, knowing that you're going to measure the accuracy on something you're going to localize with error itself. So it's not that obvious also, but this is usually what you do. So this is an example where you have two images to be registered. Here, in order just to check the registration workflow, I would say there is some beads which are added that you can see here as black spot in the EM image, and here as a white spot in the LM image, okay? And the only parts which are used for the registration are this white spot, which are also black here. And then you look at this, the discrepancy, I would say, of these points that you didn't use to say, okay, was it good or not good? And what you can see here is that, okay, in this part, for example, this is some kind of very not good. So maybe you could think there is some distortion, for example, and that you should maybe warp your image in order to correct that. And this is one way of doing that. Another way to check for the accuracy, which is basically, in that case, ignoring these points that will be just used in order to check if the method is good or not to measure the error. But basically, what you do here, you use everything that you know to be in common, okay, and you just repeat the registration by removing one point, one after the other one. So you do it with only, like here there is 7 points, so you will do the registration on 6 points, and then you will remove this point and do the registration on the other 6 points. And then for these points that you are supposed to know where they are, once again with the localization error, you will then construct this kind of graph, which will give you the number of registration. Sorry, the registration error that you have measured on the points that you didn't use, and the number of registration which had an error below this error. So for example, this kind of graph is with such as 80% of my registration were under 0.5 micrometer, so I can say, okay, I have a resolution of 0.5 micrometers for my registration. But as you can see, this is giving you a global number, and okay, here it's maybe more important, and you will never know that it's more important if we didn't have the bits. Another approaches that we have developed, which is basically based on only on statistics and not using any points, so really using the mathematical modeling of registration by itself. It's the possibility to actually estimate on any points what would be the area where you can find the points, for example, at 95% of accuracy, and this is oriented, so meaning that for every point on the image, so this is an error map, meaning that it's giving you the expected error for every point of your images with some scale, so you know where you will be more accurate, where you will be less accurate, and this kind of thing. And just to show you quickly, because it's also explaining you a little bit more about registration by itself and the way to modelize it, and also how it works for the registration error. So you can think also about registration in, for example, if I was in 1D, I just want to estimate the link between one position in X and one position in X, but in the other image. And okay, if you had, for example, an image which was linearly linked to the other image, meaning, like, for example, you just have maybe a rotation and a translation of your image, I would get something like that, meaning that I can actually fit perfectly a transform that would be modelized like a rotation matrix and a translation matrix, and that will predict exactly for any other points where I didn't know the answer what would be the transformation. Okay, and you get this kind of thing. So this is something that you know, this is linear regression, and this is one way to solve the registration problem, just trying, once again, to find the best line, the best transformation fitting this point. As we just said, in real life, you don't know exactly what are the position of points, and you rather have something like that, like points with noise. So when you will fit a line, then you will never find the green good answer. You will find another line for which is the best line fitting these noisy points, which is the white line here. And this is a transform then that you will compute, which is not the good one, which has some error. Okay, and the thing, and this is some kind of intuitive, is that because you're doing some kind of linear regression, actually, you can actually predict and measure what are the errors using these linear regression tools, and actually give confidence interval for any points on the image. So that's what we did here. And basically, now you cannot know what is a good green line, but at least you can guess what was the different solutions to find the weight lines, and then use this information, for example, to quantify the intensity with different segmentation shape or different position of your shape in order to get a more accurate measurement or a more representative measurement. So you know that. And just because that was also something mentioned by Daniel, whatever the task that you do, you need some knowledge also about what's happening. So here the knowledge is about the kind of transform that can happen to your sample. And sometimes you can say, okay, my sample has no reason to deform a lot because it was, for example, cryogenized, so it's frozen, so there is no way it would move a lot or the sample a lot. But you can also say your sample is going to deform because of the different process that will happen. And if you do that, then you will try, instead of trying to fit a line, you will try to use something which is called a non-rigid transform. Okay, on an image, it's this kind of transformation. And trying to best fit the images. The only thing is that there is plenty of different solutions, okay, to fit this kind of same points, and that's where it's super important to better understand what is happening to your sample, which kind of deformation you should apply, and which kind of I would say basis would be possible. And one thing also I want to announce on this slide is that, okay, so here in that case, if you look at these points that, okay, I knew where they are, these are my fiducials or the points that I could recognize. So for my non-rigid transforms, they are perfectly matching now, okay? But if you look at any other points, this is completely crazy for them. And this is actually happening also in real life when you do registration. Most of the time, because you don't have any way to check what's happening in the other place of your image, it's very likely that you will have a bigger error with a non-rigid transform, in particular if you did not need that. So I'm going to just mention that there is we have been developing some plugin specifically for correlative registration, and there is more option now in a new version, which is in a beta version. This is implemented under NICE software. And I'm just going to go quite quick, so we will have time for the France by Imaging presentation, maybe also, and some questions. But what I want you to maybe keep from this is that, of course, so, okay, there is some interest in doing multimodal imaging, and in particular, correlating this multimodal imaging, okay, because this is sometimes a question also, do I need to correlate them? And yes, there is reason why you want to correlate your multimodal imaging, either Either for bringing together information that you could not bring together otherwise, or for checking a new modality, for example. And the thing also is that you will have error in any case in your registration, so it's good to know what is the needed accuracy in your specific question, and usually just checking visually is not sufficient. We've seen that there is more and more deep learning coming as part of the different workflows. For some problems, sometimes it's maybe a little bit, yes, I would say overreacting, because sometimes a classical metric would be okay. And what I would say, for example, in some of the paper I've shown you, there were no real comparison with other methods. It was just, okay, it was not quantified as better than other more classical methods, for example. And because all of this, I mean, image registration by itself, you cannot work, for example, as a computer scientist on your side by yourself, because you need to understand exactly what are the modalities, what is happening also to the sample, and you need to communicate in both ways, because it's also important for people doing the experiment that they know what would be possible for the registration, if they need to add something, and what would be possible to be done. One thing also of importance, and that's also something that for now it's not easy to do with deep learning, is to estimate the confidence in your registration, because once again you don't have any ground truth. What I've tried to show you is that if you use mathematical modeling for the registration, okay, you will not have maybe the best result, but at least you will be able to estimate the result. That could be of importance in certain questions. And I just quickly mentioned you a software that we could talk about later in MIFOBIO if you are interested in. So with that, I just want to mention one last thing, is that it was mentioned by Daniel as well, but whatever, if you use deep learning but also normal methods, I mean, we say it's super difficult to validate. We don't have a lot of data set. So now there is a new network, I mean, new like 3 years ago, minus COVID. And basically, the purpose is really to bridge people from preclinical medical fields and biological fields around correlated multimodal imaging. And for now, there is a call for multimodal data set with some ground truth. Okay, we know that it will not be perfectly ground truth, but the purpose is, yes, to create database in order to train maybe network, or just to test or develop new methods to help people solve this problem. So with that, I would like to acknowledge a lot of people from Nantes, a team from MicroPCL, and collaborators from outside Nantes and some other in Nantes. Okay. So with that, I will stop and get some question and maybe.
All right, thank you very much, Perrine. Are there any questions in the room? Don't be shy. Oh, hang on. No.
Yeah, thank you very much, Perrine. So one question I have is about really where do you see the major challenges now in terms of image registration? What is really the thing where you say, okay, if I had now 2-3 PhD students, this is where I would go. This is where you expect still a major improvement with respect to what you have done and what the field has done, and which is absolutely extraordinary already. So
what I would say is it's maybe to, as you say, there is a lot of different development and things like that. What would be, a lot of them are not usable also as they are, and they are not taken into account also some difficulties that we have, like usually the volumes that we are dealing with, in particular with correlative like electron microscopy, are very huge, and it's not that easy to use. And also which is related also to the difficulty, and what would be helpful is that the different problems on the images are different in every new cases. You have a different new kind of content of your image. So it's not like in medical imaging where a brain is brain, and you have classical modalities of imaging. It's every time it's super different. So maybe trying to find a way for people to find their way, or maybe if you use a training network just to train easily on new data set with a limited number of images, for example, based on the other one, would be useful for people. Just used anything known to get new train network.
Perhaps related to this, in deep learning, what we have seen, so deep learning for segmentation, so this for in particular for cell segmentation and nuclear segmentation, so something which was really surprising a couple of years ago was that the generalist networks, is to say networks that are trained on all sorts of modalities, they're extremely robust, they work very, very well. Yeah. Even though this is clearly something 10 years ago, nobody would have done this. Everybody would have said, okay, for every modality I need one particular algorithm. And so this really came as a surprise. Yeah. Maybe this is the kind of attack angle where deep learning could really have something in registration too.
And you're mentioning, but what you're mentioning for the super generalist, if you're mentioning the data ball, for example, or but they are all one nuclei, or I mean, it's a large panel of modalities and a large panel of, but it's still the same kind of biological object. So, yeah. But yeah, that could be that indeed for sure an approach, but then it means a ton of data. So that's why there is this kind of call then in order, yes, maybe to try this universalist thing. And one last thing I didn't mention also is that one problem which is not solved yet is when you go for warping or non-rigid registration. Today there is no validated way, I would say, to estimate the error of what you've done. And I guess that would be the same question if you use deep learning also, so I don't know.
Thank you, Perrine. I'm thinking about, I'm not completely in the field, but is the simulation of the transformation when you have correlative imaging can help? Because it seems easy to simulate the transformation, yes.
You mean to create data set or to
typically if you have two images that you want to make the correlation, maybe you register first and then you apply a lot of data augmentation. In fact, we have what I mean, yes, yeah. By simulation of all kind of transformation, geometrical transformation.
So, what would be the purpose and to help to get a better, not to train a network, or to help to better...
No, to train the network.
To train the network, yes, because indeed, but that's also something you could have a basis of... I was mentioning, but I could not find, I tell you a nice reference for this, it was in another field, but some people try to do actually that, learning, as I said, the transformation itself from a basis of transformation from so the results are not like miraculous, but this is maybe an attack angle also that could be of interest, and
all right, thank you very much for your questions. I think, yeah. Hello.
Okay, thank you very much, Sophia, for chairing the session also. I know this was a first experience, great, and you kept everything on time, so thank you very much for this. And I just wanted to say, so initially I said that Edouard Bertrand would present France Bioimaging now, but then we were thinking that it would be better to have this after the common session, so just before the lunch break, so that we have everybody in the room actually. And so yeah, so this is again a small change. So Edouard, if you don't mind, it would be better to have the presentation after Martin Weigert's talk, and so just before lunch. Thank you.
fait, c'est celui de la conférence, c'est pas celui de Daniel. Hein. Celui de qui? Celui-là, Daniel m'a dit que c'était celui de la conférence.
Ah bon? On cherchait partout, le gars m'a dit qu'il en avait pas. Bon,
bah écoute, mais maintenant il y en a.
tant mieux.
Non,
Ah, c'est la pile, hein?
Ouais, non, c'est juste la pile. Hein.
Elle est là. On va en racheter 4, là on est parti en racheter 4. Au moins on est dit on est tranquille.
Oh là 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 là.
Ah ouais, ben.
Alors en plus, ça se trouve pas sur la place privilégiée.
Non.
Ben oui, si,
Il va boulanger, il y a un collègue là, qui va boulanger chez le boucher.
Ok.
Un peu plus loin pour essayer d'en acheter 4 avec des piles de rechange.
Okay, là, là. Oui, les piles encore, ça se trouver, mais...
Et en fait, il y a beaucoup de problèmes avec les Mac, tout ce qui est compatibilité derrière. Du coup, ça va avec les PC.
Ouais, ben oui. Bon.
Ah non.
Ça va? Oui, pour...
Cette petite doc, c'est ça. Donc là, c'est le pointeur, c'est là.
Le pointeur, j'ai laissé là. Le micro, je peux le laisser là, mais ouais, c'est bon. Je l'éteins peut-être.
Abelite, qu'on remercie également. Voilà. Et donc je remercie tous les participants, c'est-à-dire tous les gens qui sont passés par la formation permanente, tous ceux qui sont inscrits comme participants, d'aller signer les registres à l'entrée. C'est très important parce que sinon, vous avez un problème avec votre formation permanente pour la prise en charge de vos missions. Parce que vous serez déclaré comme étant absent à la formation. Donc il faut que vous ayez signé le registre sur tous les jours. Donc pour vous simplifier la vie, on vous les fait signer maintenant. Maintenant, je propose à... il y a Anna qui voulait... Vas-y, Anna.
Oui, bonjour à tous. Je voulais juste vous dire une information, or I will tell it in English maybe, that we decided to prolongate the voting for the posters till Sunday, so because there's really a lot of posters, and if you want to see more and you want to discuss more, you still have the chance. The posters will stay there and you can still vote till Sunday, so please do. Thank you.
Bonjour à tous. Non, je suis là pour vous présenter le Fab Lab que nous avons créé depuis 2016, je crois. Une initiative de 2016, je crois que ça fait troisième représentation. C'est le groupe de travail du RTMFM qui va animer ce Fab Lab. Donc il est à la sortie de la salle, ici à droite. Alors il est composé d'experts un petit peu dans tous les domaines, d'impression 3D, d'électronique, de pilotage. Enfin, si vous avez besoin de l'utiliser, ce Fab Lab, il est libre, il est en libre service. Nous avons des imprimantes 3D, nous avons beaucoup de microcontrôleurs, d'appareils à souder. Vous pouvez venir quand vous voulez, en fait, c'est open. Nous avons aussi quelques ateliers que nous allons animer. Voilà, là c'est une photo de ce que vous avez en sortant à droite. Donc le libre service, imprimante, soudure, des fiches tuto. Vous pouvez donc, même si vous n'avez pas d'idée, vous pouvez venir prendre une fiche, l'ouvrir et réaliser ce qui est indiqué sur les fiches. Nous avons des composants électroniques, nous avons tout pour vous aider. Le but c'est de faire des interfaces pour la microscopie. Alors on a 3 ateliers cette année, un qui est progressif, animé par Brice Roncin et Thierry Legout. Donc 3 niveaux progressifs, vous avez dû les voir, ils sont sous le numéro A080. Microcontrôleur, nano-ordinateur et objets connectés. Ensuite, on a aussi un atelier qui s'appelle A084, animé par Ludovic Lecomte et Brice de Tailleur. Et là, ce sera pour l'impression 3D, dessinée, paramétrée et imprimée. Donc vous pouvez venir, et si vous n'avez plus de mug, on pourra toujours en imprimer un peut-être, je ne sais pas. Et enfin, un challenge et une devinette. Donc le challenge, ça va être un petit montage comme ça, avec un microcontrôleur. Et des questions, il y a 3 questions. Si vous arrivez à les résoudre, vous gagnerez un kit. Un des kits que nous utilisons sur l'espace, c'est-à-dire une Raspberry, un Arduino, et voilà, pour vous amuser chez vous. Moi j'ai commencé comme ça, c'est un ami qui m'a donné un jour un Arduino, et depuis ça fait 10 ans que je suis dedans, quoi. Et enfin la devinette, voici une image, il va falloir que vous nous dites ce que c'est, sauf celui qui l'a créé, qui est dans la salle d'ailleurs. Et si vous arrivez à trouver ce qu'est cette image, bien sûr celle de gauche, vous gagnerez aussi un petit kit de développement. Voilà, je vous remercie.
Merci Christian. Et donc cette année, on avait initié déjà en 2018 quelque chose qui était autour de l'optique, Optic Lab, et là c'est devenu vraiment un groupe dont Laurent et d'autres se sont occupés. Donc je vais laisser vous présenter et là aussi vous expliquer un petit peu l'esprit.
Merci. Bonjour à tous, good morning. Donc le parcours Optic Lab, l'idée c'est d'avoir des ateliers qui normalement sont aussi en libre service. Ceci dit, vous avez de la chance, c'est en première mondiale au MIFOBIO cette fois-ci. Donc du coup, il y a sans doute des gestements, on vous conseille quand même de venir pendant les créneaux ateliers. Donc on a 8 créneaux sur lesquels on peut vous orienter, et puis après ça, vous êtes libre de pratiquer ça vous-même. L'idée c'est quoi? L'idée c'est d'apprendre des principes de base d'optique et de métrologie. Et on a associé à chaque fois, on a créé 4 parcours. Vous voyez les thématiques, plan conjugué, illumination échelle optique, diffraction et résolution, aberration chromatique, etc. Et pour chacun de ces parcours, il y a une activité sur une table optique où on comprend les principes de base de l'optique et suit après ça sur un microscope un exercice de métrologie. Ça peut être faire des point spread functions, ça peut être aligner des anneaux de phase, ça dépend de la thématique. Donc les deux sont liés. On a été un certain nombre de gens à préparer tout cet atelier. Amaury, Pierre, Sophie, Aurélien, moi-même et Damien. Et je voudrais avoir une pensée particulière pour Damien, qui, pour des raisons de santé, ne peut pas être ici, mais bien qu'il savait qu'il ne pourrait pas venir, a continué à produire du matériel et des documents. Donc on est très reconnaissant et on lui souhaite un prompt rétablissement. Et on est aussi très reconnaissant à Thomas Gilbert, qui va du coup, au pied levé, prendre la relève et nous aider à encadrer ces travaux pratiques. Alors très rapidement, voilà. Donc, comme je vous l'ai dit, il y aura à chaque fois, pour chaque parcours, vous commencez normalement à travailler sur une table optique, à faire quelques exercices pour comprendre, aussi avoir des définitions et des principes de base. Et l'idée après ça, c'est d'aller sur un microscope et de voir, dans la pratique quotidienne, plus souvent en tant que biologiste, comment ces pratiques de base vous permettent de vérifier la performance de votre microscope. Donc, on a aussi six gros microscopes qui nous ont été généreusement donnés. Je voudrais remercier quand même Nikon, Olympus, Zeiss et Leica qui ont tous accepté de donner des systèmes et de les mettre dans une même pièce, ce qui est très sympa. Et puis enfin, on a aussi un peu de matériel pour ceux qui voudraient apprendre à préparer des échantillons pour la métrologie, notamment à faire des lames de billes ou à préparer des lames pour tester du vignettage ou du shading. C'est aussi le but de ces ateliers, c'est de vous donner, c'est pas très compliqué, mais il y a des petites astuces. Voilà, l'idée c'est de vous donner les astuces pour préparer vos échantillons. Et il y a enfin un défi. Le défi, ça sera de coupler au mieux deux lasers dans une fibre. Et je ne sais pas ce qui a gagné, mais le plaisir d'avoir été le meilleur et d'avoir réussi. Voilà. Il y a un prix. Okay. Voilà, donc on espère vous voir nombreux. On a 8 créneaux. Mais encore une fois, vous pouvez aussi travailler au dehors des créneaux une fois que vous avez les instructions. Ouais.
Donc, pour ce défi, on va faire un fonctionnement du type maillot jaune. C'est-à-dire que tous les jours, il y aura le plus performant du jour. Et au fur et à mesure du déroulement de MIFOBIO, qui dure un peu moins qu'un Tour de France, mais qui est à peu près aussi intense, avec un peu plus de bière à la fin. Toujours est-il que les plus performants chaque jour seront identifiés et on verra à la fin. Donc il y aura parmi les prix, il y aura aussi des défis qu'on va vous présenter cet après-midi. Donc c'est des petits challenges pour se challenger entre nous. Voilà. Et donc le banc, il est prêté par Oxius, qu'on remercie également. Donc maintenant, on va repasser au reste du programme. Je remercie les orateurs de ce matin qui ont déjà initié les deux premiers modules, le module 1 et le module 3. Donc maintenant, on va passer à la suite du module 3 avec les intervenants en plénière. Et cet après-midi, donc vous avez d'abord un workshop, une session à la fois workshop, table ronde en début d'après-midi, et après la pause, à 16h10, on se retrouve donc vers 16h pour ne pas prendre de retard. Il y aura aussi une petite présentation, donc on va dire 16h, et pas 16h10. On se retrouve ici pour la session de l'après-midi, avec le module 2-3. Donc il y aura une partie, à nouveau, à Porquerolles. Pour indication, le Optic Lab, il est du côté de Porquerolles, donc de l'autre grande salle. Et puis une partie ici en Méditerranée. Et juste en fin tout de suite, là, en fin de matinée, on laissera la parole un petit peu, pour une dizaine de minutes, à France Bioimaging, à Édouard Bertrand, pour qu'il présente ils sont en train de développer une approche autour du traitement de données, donc associé au concept de ce module, du module 3. Voilà. Eh ben, je vous remercie, et donc place au module 3.
Okay, so now we start the next session and we have the pleasure to hear Dr. Loïc Royer. So Loïc is a group leader at Atlas Initiative in Chan Zuckerberg Biohub, based in San Francisco, California. So Loïc obtained master degrees in artificial intelligence, specialized in cognitive robotics, followed by PhD in bioinformatics from Dresden University of Technology in Germany. So Loic works in a pluridisciplinary team of computer scientists, optical engineer, and biologists. And the goal of the group is to build a time-resolved multidimensional atlas of vertebrate development using zebrafish as model organism. And to reach this goal, they design novel state-of-the-art light sheet microscope and design deep learning-based image processing and analysis algorithm. So now we talk about self-supervised deep learning from fluorescent image and AD image viewing with Nepari. So here we go.
Thank you very much.
I'm not
Okay, it's very rock and roll. Okay, great. So thanks a lot to the organizers to invite me today to talk about my team's work. It's a very nice meeting. I've never been here. I've done most of my career in Germany and the US now, and so it's very nice to connect a little bit with the French community. So today I will talk mostly about our work on deep learning, image processing in general, and basically let me learn to use this tool perhaps. How do we do that? It's not working. Which button do I need to press? This one?
Yeah, I think it's
No.
yes,
The middle one? Oh no, I mean to move from one slide to the.
Okay.
Oh, it's not doing. Ouais, je l'ai mis. Bon, il a pas. Ouais, il est là. Non, ça marche pas. Bon, c'est pas grave. Not a big deal. Okay, so great. Thanks for the help. Basically, so where do I work? So my team is at the Chan Zuckerberg Biohub, which is a non-profit research institute in San Francisco, which brings together Berkeley, UCSF, and Stanford. And actually, by the way, we're kind of hiring right now, postdocs or and actually in particular right now I'm hiring an optical engineer. I will talk about it again. It's a staff position, so come to talk to me if you're interested. This can move very, very fast. But anyway, so this institute where I work is a very nice place because we bring together the three universities, and we have a very nice research program involving infectious disease, but also a quantitative cell science. So my team there does three things. A little bit crazy. We're trying to do three things at the same time. The reason why we do computational microscopy and biology at the same time is because we're trying to come up with innovations that are synergistic in nature, and trying to bring together these three disciplines to push the state of the art and get some, in fine, get really interesting biology answered. So essentially, we build a Ferrari, which is a combination of hardware and software, and we drive it on the racetrack trying to get to some biology, right? So that's what we're trying to do. And right now, most of my team is working on this, and everything else is old news. So I'm going to talk about old stuff, but I mean, in the next months and within the next year, you'll be hearing about new stuff coming here, which is mostly actually biology, leveraging all the technologies we've developed until now. So, as I said, right now I'm hiring an optical engineer to build very, very advanced light sheet microscopes. We have already done some work in that field. We have built some very interesting single objective light sheet microscopes. And so if you like microscopy, if you like optics, if you like light sheet microscopy, if you know about our work, please come to talk to me, and we have a job for you perhaps. So this is an overview of the projects we have currently, kind of published work, everything from automatic fish feeding, because we use zebrafish as a model organism for our research. We have built this single objective light sheet microscope, and then we have a bunch of projects involving deep learning, image processing, image analysis, among other things. And so really today I will focus on that. That's the organizers asked me to talk about artificial intelligence, deep learning applied to microscopy. And so let's get started with something that's a little bit of an old story already, but there's some new developments. So after my talk, Martin will present his work, and of course you might know Martin's work from his PhD, CARE. And this was a very successful project. He did an amazing work there. And the results were very spectacular, but of course the problem there was that we had to have the ground truth to learn, to basically be able to learn, to teach the networks to improve the quality of the images that we had. So what are the limits there? I mean, deep learning has been very successful in recent years. There's been a gazillion different applications, all the way from segmentation, tracking, image restoration, and more, super resolution, you name it. But there's perhaps a few limitations, or at least we need to understand when we have a very powerful tool what its limits are. And the limits are far, but they're nevertheless there, and so it's important to be conscious of that. So we recently we wrote this perspective in nature methods that kind of went through all these things, and there are lots of applications, lots of promises, but also pitfalls that one has to be careful with. And one thing that people like to see oftentimes is, and it's kind of probably the best figure in that paper. Is this figure that highlights a few aspects of deep learning that are important when applied to images, or at least in the case of restoration, is if you have an image that is degraded like this one, you can barely see what is written there. The importance of the training data set is key, right? So if you have the right training data set, you might be able to recover the true text. If you eliminate some letters from that, you will actually start hallucinating, or actually replacing the real characters with confusions. So it will get confused and put a Y here and some weird symbol here. And of course, if you train on the completely wrong data set, it won't work well, right? So you will have, you kind of see where the signal is, but you can't really tell what really is there. And so what that means is it's really important to actually train with the right data, right? And sometimes in microscopy it's not possible to acquire, your ground truth, the perfect image that you can use to train the network. So that's an issue. And there are limits to the idea of let's train on everything available, because the truth is, if you would train a network on all possible images, it's as if you train it on nothing, because essentially you're not specifying any prior. You're not explaining to the network what is the space of images you care about. So although augmentation and pre-training on natural images has been successful in some cases, there's a limit. There's a fundamental limit to what you can do with these approaches. So having said all this, I mean, what's remarkable, and sometimes something I have to point out is, although you have to have some training data, you are able, in most cases, to discover something even though you knew something beforehand. Sometimes people say, well, but you're training on, you have to have this ground truth, so can you really discover anything new? And the fact is you can, because although you might know the letters, you might not know the word. So training on all, using a training data set that consists of all letters, for example here of the Latin characters, you actually can recover a word that you might not know. So this analogy applied to biology means that you can use deep learning in your training on ground truths that you have, and still discover something new, because it's oftentimes not just the atoms that you need to discover that you might know, the elementary pieces, but what you need also, what you can discover is how they fit together. So definitely interesting to think about this, and so the key question when I started in San Francisco. When I was still thinking a little bit about these ideas, the key thing was, like, can you train networks without any ground truths? What if we don't have a way to get these clean images? What if it's just impossible from an experimental standpoint to get these images? And so I was lucky that at the time, at the BioHub there was a very talented mathematician who was dabbling in biology and genomics and other topics there. And together, I asked him, can we do this without, can we train networks without a ground truth, and can we do image restoration without ground truth? And so we were thinking about it for quite some time, and then we basically came up with a very nice idea inspired by a previous paper called Noise to Noise, and we realized there was a way to do that. And so this was quite of a nice development. By now, there is actually a gazillion variants of this idea. I think there's about 150 different, variants, and as much citations on this. So it's pretty. It started a whole kind of, together with the noise to noise and some other papers. This all started a new kind of trend in the deep learning community. And so in the past years, since this paper was released, we've been thinking hard about how do we share this with the community in a way that is very practical. And so we made a thing. We made a thing with two members in my team. Chan Solak, who is a software engineer in my team, and Hirofumi, who is a postdoc. So we basically put together a very comprehensive software. I mean, we put quite some work on this, and it's extremely rich in features. So essentially what this tool does is it's a UI, fully standalone, so you basically download it, you open it, you drag and drop your image, you can crop, you can redefine the meaning of dimensions, you can do all sorts of things to facilitate denoising, and we have things like pre and post processors that allow you to apply transformations through your images that will make denoising easier. We have, I think about 15 or so different denoising algorithms in there right now, and we have 20 more planned. And the thing we learned while doing this is there are many kinds of noises and degradations in images, and so there's not one algorithm is not going to make it. You need a multiplicity of algorithms. And the ability to tune a lot of parameters to be able to get to the best result. And thanks to the mathematical theory behind noise to self, we have actually a way to tune all these parameters automatically. And so we will release this software soon, but eventually the goal is to have a one-click where essentially the software will automatically figure out among all these different algorithms and parameters which one is the best for your data. And in some cases, what we've discovered is that you don't even need deep learning to get really spectacular results. And in some cases, the denoising can be done within a minute. And so this is a data set from my lab where, this is essentially what some of the imaging we do routinely. This is actually not even light sheet. And so we can do the denoising pretty fast. I think for this image, it was below a minute. And the results are really good. And so what that means is that in many cases you do not need to use very complicated techniques. Sometimes you have to. Sometimes the noise is really complex and difficult, and then it's worth to use very complex methods. But it turns out that a lot of classical algorithms and variants of them that we have developed can be auto-tuned, and in some cases they're so fast that there's really no point in using anything else, and the performance is oftentimes good enough. So there's a whole spectrum of things we can do, and we have, when we release this on Monday, we will have a lot of use cases for you to try. Essentially, we go through different examples of data sets that you can try. And see how see for different kinds of images from different kinds of microscopes or imaging systems, we can actually get really nice improvements. And so I will be here the whole week. And so if you're interested in chatting, if you have images that are noisy, you want to try our software, you'll be able to do so. And on Monday it will be available. And in some cases, as you can see here, the denoising can be quite spectacular. And again, you don't need any ground truth. You just drag and drop the image, and everything is automatic. So this is kind of probably the last thing that I will do with denoising. It was right now denoising is a little bit of a hobby of mine. As I said, most of the team right now is working on biology and microscopy, one collaboration that I've been doing, with Hero in my team with Manuel Leonetti and Keith Sherrills at the BioHub is this very nice Cytosoft project. So let me explain what this is about. So Manuel is a group leader at the BioHub, and what he's been doing is systematically tagging endogenously human 1,000 proteins in human cell lines. So essentially, labeling with GFP one protein at a time in a way that preserves the endogenous expression levels and makes it such that the localization of the proteins is actually very accurate. So this is the most accurate image-based localization profiling of proteins in mammalian cell, in human cells done to date. And so it's a beautiful data set. I will not go into the details of how it's acquired, but this has been the work of Manuel's team for the past 4 years, and it's a beautiful work. And so what we decided to do together with Manu is, can we try to make sense of all these different localizations that exist in human cells? So you see here a sample of different cells imaged on a confocal microscope with different localization patterns. And so you can think like, what's the complexity behind this? What are the kinds of localizations that are there? And can we just understand that as a baby looking at the world? Can we just not impose our own conception about what is a nuclear localized protein, what is a cytoplasmic localized protein? Can we try to discover something from the data on a purely unbiased and kind of data-driven manner? So when you think about it right now, there is basically these amazing technologies which actually we are also dabbling in right now, which is you take cells and you can dissociate, you can take tissues, you dissociate the tissues, you get cells, and then you can do single cell sequencing. And what happens, and then you have for each cell a vector that describes the expression levels of each protein, and so that's beautiful. And so you can do this with essentially RNA levels in each cell. And if you think about microscopy, it's a little bit of an issue because we get images, and then these images are not really vectors, so it's difficult to do all these fancy UMAPs that people do in single cell sequencing, because okay, you get lots of images for each protein localization, but that is not really conducive to analysis. So what we need really is not images, but turn these images into vectors, right? And so we need a way to turn an image, which is a complex object, lots of data in there, which pixels to pick, which features to pick. It's a complicated thing. So the the way you ascribe meaning to vectors is really you say, okay, I have 2 vectors, I can ascribe a distance between them, or I have a different coefficients made by vector, and I can ascribe meaning to each one of these values. And so the classical approach typically is you take an image and you have maybe a bank of filters or pieces of code that extract features from that. That's very typical, for example, in Cell Profiler, and you can extract things like size, shape, basic textures. And then once you have these vectors, you can do classification. You can also do clustering, which is a little bit less, a little bit more data-driven. And more recently, of course, with deep learning and machine learning becoming very popular, people are starting to learn these features. And then the question is, how do you learn them? What is the thing that drives the learning? But you can do then classification, and again, you can, of course, do clustering. And really, the benefit of doing learning for both features and then using unsupervised approaches for clustering is that you basically, humans don't impose anything on the data. The data just tells us what it is about. And that's what we want to do. We want to let the data speak for itself and basically have no human input to the data. So can an algorithm at initio make sense of images? Can we just let an algorithm tell us what these images really say? And in a way that in the same way that we would look at these images and make sense of them. So when you think about information in images, it's a difficult topic. It's a complex topic. Here's a question: which one of these 3 images contains the most information? And so the right answer here is another question, which is, what does it mean? What does information mean? And really, even better answer is that it depends. Information is relative. And that's a very important thing, is information is not an absolute notion. It's really relative. And so the way that information is relative, it's relative to the encoder-decoder frame of reference. So you can think of it as, you need something to encode the information into some distilled representation, and then from the distilled representation, you should be able to recover the original image. And if you have such an encoder-decoder scheme, then you can speak about information in a relative fashion, relative to that pair of encoder and decoder. And if you actually have these little images of triangles, then you realize that actually the content, the information content in images can be very little. I mean, here, if you want to encode the position of a triangle in an image, you just need 60 bits of information, you know, if you don't have colors for the triangle or so. And so you know, what typically happens is you basically you can essentially, what an encoder-decoder can do for you is it can, if it's well designed or well chosen or well trained, it can actually extract that important information. And that's again a relative thing. You know, what is the important information? What is the information you care about? And so that these are all the key things. And so in the case of biological images, really, how do we come up with that? And so here's where deep learning really has helped us dramatically, is that it can help us discover the best autoencoder, which best is also a relative thing, right? So how does that work in practice? So we take our fields of view coming from our microscope, we crop the fields of view into small tiles, and we feed them to a neural network which is, to start with, really naive and doesn't know how to encode and decode. But we force the autoencoder to be able to create a very small vector, and from that very small vector, it has to be able to recover the original image. So it is forced to be able to do that compression and decompression if you want. And so at the middle, that middle place here, if I manage to, this vector here is very small compared to the size of each one of these images, and so the network is trained to be able to come up with that kind of distilled and compressed representation. And so you can do that, and lots of people have done that, and you know, also outside of biology and microscopy, with other kinds of images. And if you do this, you can actually get these vectors, and then the reconstruction is pretty good. You know, you fit in the image, and you can actually get an image that's pretty close. But then if you try to take these vectors and see what they tell you, you know, if you try to do dimension reduction using UMAP or PCA or TSNE, the maps you get are not very informative. So it seems that, you know, this is not really doing anything useful for us, right? And what we discovered is that there's a very simple thing you need to do to actually make this work beautifully, is that if these vectors, then you ask these vectors, in addition to being able to reconstruct the images, if you also ask the following thing, if you ask that, okay, I need to be able to predict from which field of view this crop came from, essentially predicting the identity of the protein from this vector, meaning that if two proteins have identical localizations, the system would not be able to distinguish them, but that's fine, because they are really the same localization. Therefore, the information contained here should be the same. So we just do that, and then lo and behold, the result is really spectacular, and so Manu was really stunned by the result, because the map we get is beautiful in many ways. Before I go into the details of what we found there, I have to say what I showed you is a little bit the oversimplification. It's much more complex. We have what we use, you know, for those that are a little bit more technically inclined. We use a VQVA2 architecture that has been modified in a number of ways, including, you know, an additional loss to be able to do this prediction of the idea of the protein, which is a kind of key thing. That's all of the whole paper is this. And we have other innovations such as split vector quantization and some other things. But these are the details. This is complicated, but the really fascinating thing is what we get from the results. So this is the map of protein localization with different, you know, human-defined categories that we superimpose here with color. But you can see there's so much structure here. And so the question is, all these groupings and subgroupings, do they have meaning? What is behind them? And we can overimpose our own preconception about what we think is what, but let's look at what the data tells us. Well, if you zoom in, complexes just pop up. So what we've discovered is that you can almost predict protein complexes just from images. So here you see the SF3 spliceosome nicely delineated. And actually, it's worse than that. You actually can see the subcomplexes in some cases, which is nuts because you wouldn't be able to tell the difference between these images. I mean, you wouldn't be able to see that the reasons why some of these images that correspond to proteins actually should lead you to the conclusion that there are actually subcomplexes there. And so if you keep looking at it, we just find other protein complexes that are well known. So we can recapitulate a lot of biology about protein-protein interactions just by looking at images. And it just goes on and on. And everywhere we look, there's biology everywhere in this map. And it's entirely discovered from first principles, just from the images. We didn't input any human knowledge to that. And so we looked quite systematically throughout the map, and we found a lot of very interesting things. And you know, in the open cell paper, which is kind of the sister paper, we have other analyses also which are very interesting. So with that, I'd like to close this part of my talk, and then just acknowledge this collaboration with Manuel's team. Many people at the BioHub contributed to this project, which is OpenCell and Cytoself. There was also this also involved the collaboration with the Max Planck Institute in Germany, and also a number of our collaborators at UCSF. So I don't know how much time I have left. The third part I wanted to talk about was Nepari, and I have a lot of slides, but I will basically maybe cut it short as soon as you guys give me a sign that it's time to move on. So when I started my lab in San Francisco, I came from Dresden. In Dresden, I was well connected with the whole Fiji team and a lot of people working in the Java world of bioimaging. But when I arrived in San Francisco, I realized that it was difficult to hire people that knew Java, and or they were actually hireable or affordable. And also, I was kind of interacting with a lot of Python folks there at Berkeley, and so I kind of fell in love with Python, and wanted to use more Python. But then the issue was that I couldn't look at my images. And looking is the first step to discovery. So we were building microscopes, we were designing image processing, image analysis software, and then we couldn't look at our images in Python or in any way that was satisfactory to me. So and so basically, I started a project because I just needed to look at my images. So I started a project with Juan Núñez Iglesias, who's a good friend from Janilia, who was then in Melbourne, Australia, and so that's why we chose the name Nepari for this project, because I lived in San Francisco, he lived in Melbourne. That was an island somewhere in the middle of the Pacific, and so basically we started this project in Python for viewing n-dimensional images in a very fast and convenient manner, using kind of a Google Maps type of way to browse your images, to pan and zoom. And since we started this like in 2008 or so, this project completely exploded. We have like 10 core developers helping develop this software. We have now more than 90 contributors. This slide is already outdated. And with amazing people such as Nick Sofoniev, Tali Lambert, Kiara Evans, Kira was the first person the second person writing code for Nepari after myself, that I hired as an intern under the recommendation of Juan. And but since then, the thing is, I don't recognize the code anymore. It's just moving too fast. And Nick has been really moving this project full time, and within CZI, who has also invested a lot of funds to. And it's about 20 people being paid by CZI to work on this project. So. It's a crazy thing to see, how a weekend project that I started with one just turned into something that like more 100 people work on it. And so what is Nepari? So Nepari is a fast visualization a library and application for n-dimensional images. It integrates with the Python ecosystem. It supports arbitrary large data, which means too large for memory, possibly remote data. It's very extensible, it's very customizable, and it's inclusive. It's community-driven. I think it's the first real open source project that I'm part of. I was always writing code with friends, but in the open, but that's not really open source, as I now realized. And then we rest on giant shoulders. I mean, the thing that seduced me so much with the Python ecosystem is there's amazing libraries that are used in science with a big S outside of biology, and so there's a huge community of physicists and meteorologists and you name it. So many scientific disciplines. They're all working together on these tools, and so building something on top of that for the bioimage community was really enticing to me, and so you can install it from if you're a Python person, you'll understand what this means, and so you can just pip install it. You can also content install it. It's also for your quote-unquote biologist friend. It's also an app you can just download. You double-click, you start it, and then you can start using it. My vision for it is eventually Nepari is the only Python distribution you will ever need. That's a little bit pushing it, but I like to push things. So how does it look like? So that's a Nepari viewer. That's the main thing about Nepari. So you have a canvas where you can look at your images. You have a layer list. Instead of lots of windows popping around everywhere, we basically have this idea of layers, a bit like Photoshop. And you can layer a lot of different kinds of things on top of each other. And not just images, also vector objects and other such things. You have layer controls for each layer. You have a dimension slider like in Fiji or IC and these tools. And it's arbitrary. You can have an arbitrary number of sliders here. So if you have a 15-dimensional data set, you'll have either 13 or 12, depending if you do 2D or 3D viewing. You can add and remove layers. You can control the canvas in terms of switching between 2D and 3D and such things. And you have a status bar, as most tools provide. So the layers are really the central thing, because that was really something we really wanted to have, to go away from the kind of tons of windows, cluttering your screen. And that lets you overlay different kinds of information, like images, of course, multi-channel images, of course, but also points, labels, and more. And we have all sorts of different layers, vector layers, track layers, surface layers, and so you can not only look at the image data, but also the result of the analysis of this data, and overlay that, and basically build interesting workflows with that. So if you look at the image layer, you can definitely look at your images in 2D, in 3D. It's literally a click and then you switch to 3D. You don't need to start a plugin for, with Martin, we worked on Clear Volume, for example, that you might know. That's a plugin within Fiji, and you have to start it, and here it's just one click and you switch to 3D. And you can turn on and off the layers, control each layer, and so on. So very, very practical. And my team, everybody uses Nepari. It's just a thing to use for us for these reasons. You can also do multiscale, multi-resolution viewing of very, very large images. You can do on-the-fly processing. If you're a little bit more, Python versed, you can do things with Dask and do on-the-fly deconvolution and skewing. Really great work from Lambert. Lambert actually made these slides for the community. So I'm just showing the slides. I mean, I modified them a little bit, but this is really Tally's work. We're all using his slides because they're so beautiful. And so yeah, we have segmentation layers, as you can see here, where each segment appears in a different color, and you can actually interactively interact with some of these layers. and you can paint, and so you can imagine how you can build interactive workflows on top of your for analysis on top of your image data. That also works in 3D. Layers work also in 3D. You have also point cloud layers, which can handle quite a lot of data, because everything in Nepari is GPU accelerated. So we rely on the vSpy library to be able to render very fast a lot of geometry. That was really something I wanted to have from the get-go, from the start. I wanted to have the highest possible graphical performance, but in a very, very easy and accessible package, if you want. So here, vector layers in 3D. So you can, as you see here, very nice work from Alistair Bird, who's very active in the Nepari community. You can overlay vectors with points, and on top of your here, in this case, 3D electron cryotomograms. This is HIV particles. Track layers, super important for us. We do a lot of tracking of zebrafish embryos from the images of our light chain microscope. So we love these track layers, of course. And you very nice plugins. So a lot of people have been contributing plugins to Nepari, and one of them is Arboretum from Alan Lau lab at UCL. Which were very, very nice, plugin where you can look at the arborescence of your tracks and lineages. Surfaces. I mean, I don't need surfaces for my work. I mean, right now at least I don't, but I'm sure someone needs that. If you work in, for example, in kind of more like medical imaging, perhaps, you can draw shapes, so you can really bring in your manual annotations and then have it part of a pipeline. So the cool thing is, the reason why I said that Nepari might become the only Python that the biologist needs is that we have integrated the IPython terminal within Nepari, so you can actually do scripting in Python within Nepari and interact with the things you actually annotate within the interface. So you can basically get the coordinates of the points that you have added on the canvas, and then do this kind of ping pong between the visualization, the annotation, and the running raw Python code and any kind of library you would want to run from there. And so, for example, Juan kind of implemented the kind of tensor board for noise to self within Nepari, which is kind of fun. So here you see a neural network being trained, and he visualizes simultaneously the result of the training of noise to self on these images, and the loss of the network during training. And so I said, lots of plugins are being developed for Nepari. It's very easy to install a plugin right now. It's very easy to build a plugin if you know a little bit of Python, but it's also very easy to install them. Sorry, I just clicked too many times. So we have a very nice interface for installing plugins. So if you want to distribute your creations, it's a great way to do so. And once you install the plugin, it's there, and then you can use it within Nepari. And we have a cookie cutter Nepari plugin, which means it's a plugin that's empty, and you can just clone that repo and just put your own code in there, and then you have your own plugin, and it's very fast. Martin has made a very nice Stardust plugin for Nepari with Uwe, and I asked him, how was your experience with that? He said it was great. I don't think he knew that I would mention that now, but maybe if we would have changed his statement. But yeah, apparently it's working well for people, and so lots of plugins out there, and many of them rely on Magic GUI to actually generate automatically UI elements from Python code, which means you don't have to learn complex UI programming. You can rely on very simple things. You can, and as I said, lots of plugins for reading images, like TIFFs and other things. You have Momero is also a plugin now. You have even a Micromanager plugin, which is being very actively developed and is moving very fast. Robert, our good friend Robert, he's definitely very active in the Nepari world now and writing lots of plugins. I think one plugin a week or something. And so of course his whole suite of open cell processing tools is there. CellPose is there from Carson Stringer in Janilia. Beautiful plugin, works very well. And then Stardust from Uwe and Martin presenting just after me. And so Finder. So that's an important one. That's Nepari animation. That's a really cool plugin to actually create animations from within Nepari using key point animation. Something that actually, for those that know the Martin's tool SPImagine, which to some extent was an inspiration for some aspects of Nepari. He had also a very nice way to generate movies using keyframes. And so we have the same thing here with the Nepari animation. You can build an animation with keyframes and basically do anything you can do by hand in terms of changing the views and turning on and off things. You can do everything from Nepari animation. So you can make pretty complex animations, like this one, for example, where you can show an image data, go from one object to another, and do rotations, and so on and so forth. So yeah, that's it. Thank you for your attention. But I didn't. It wasn't too long. But yeah, thanks a lot again for the organizers for your attention, and I'm just very happy to answer some of your questions.
Okay, thank you, Loic, for this good presentation. Now it's time for some questions.
Hey, Loic.
Hey, man. How are you? Hi, Seth.
I really like your work, you know that.
Thank you.
I really like the idea of the self-supervision to bring the similar proteins close to one another.
Thank you.
Eventually, what you get from that is localization patterns, which is also what you get when you use annotations. So the question is, can you get something more than that? Can you get the discovery that is beyond what you would get by localizing these proteins to what organelles they are localized at?
So I'm not sure I understood the. Sorry,
So for example, the Human Protein Atlas, the challenges there is trying to localize a protein to the specific organelles that it's localized to, right?
Yes, yes.
The question is, and what you show here is something similar. Just I mean, you don't give it any information. Nothing is supervised, which is fantastic. It's really fantastic. The question is, can you exploit the unsupervised, not giving it a direction, to actually get something more out of that?
More than the images themselves have, or
more than saying this protein is localized here, and here.
I think fundamentally, the loss we use during the self-supervision is really aimed at localization. I think interesting things we want to do next is, for example, you perturb a cell and then basically you see how, for example, a protein moves from the nucleus to the cytoplasm or back. So in the revision of the paper, we are considering not that, but we're considering things like, for example, can you use a data set from a completely different lab or different microscope, and can we basically infer localization? But I think, I think it's really squarely working on the localization. I mean, we can maybe chat offline. I must say, like, we can maybe discuss that. But I think localization is the thing. It's doing that very well, but maybe only that very well. Perhaps we can chat if maybe I'm interested. Maybe you have ideas of what you could do with that. So maybe we can chat.
Yeah, thank you very much. So perhaps also on the same topic, on the protein localization, so you showed clearly that if you use an autoencoder and then you use the latent space for just projection and then cluster analysis or whatever, this doesn't work very nicely.
Yeah.
And then you complemented this by the prediction of the protein identity without specifics on the localization.
Exactly.
But one question is, how much does the latent space actually help? You could also say, I'm just predicting protein identity, and then work with the representations that just serve as this. And so how would this compare then to
we tried
using both actually?
So we tried to literally just go straight to the prediction, right? So and what's interesting is it works better than if you don't have. So the key thing is that loss, right? That additional prediction. If you have that, things are good. If you don't have that, things are bad. But if you have all the features we added, then you have the absolute best performance. And everything that you remove will decrease the performance. But if you remove that prediction, it just collapses. So everything we did helps and is part incrementally of the solution. But there's one piece that holds it all together. But you can omit the decoder. You can remove the decoder. The thing is, if you just have an autoencoder, the representation that you find is one among many. And there's so many ways we force the system with that additional loss to pick the one that is the best for our purpose. And that was kind of the key insight of the work.
Okay, thank you again.
Thank you.
Segmentation for microscopy. He did his PhD at Max Planck Institute of Molecular Cell Biology and Genetics in Dresden, Germany. And Martin recently joined a PFL. So now we'll speak about microscopy image analysis with machine learning. Here we go.
So, hello everyone. I'm pretty excited to be here. It's my first MIFOBIO, and I think it's the first talk in 2 years that I have to give not in front of a computer sitting at home. So that's exciting. And of course, I brought my clicker, which I forgot to change the battery. So first thing, I just realized it didn't work, but Rainer Heinzmann on the back helped me out. So that was kind of a stressful entrance. So yeah, thanks again for the organizers for inviting me. And today I will speak mostly about the work that we did in the last 2-3 years. So it's going to be stuff that you probably already know, but I still want to mention it. And at the end, a bit more recent work. And in generally involves using machine learning, so deep learning in particular, to solve certain problems that appear in microscopy image analysis and the microscopic acquisition process in itself. And I always like to start with just a short slide that shows the variability of images that you can get from microscopes. Of course, I'm preaching to the choir here. Everyone is doing microscopy, so everyone is aware of the beauty of those images. But I just want to show that the variability of the modalities is quite extensive, right? It ranges from fluorescence microscopy to H&E stained histopathology images, bright field images, like in this case, electron microscopy stacks that can be gigabyte-sized. And as well light sheet microscopy that creates 24-hour recordings in 3D over time. So it's a huge span of data and a huge span of different modalities and different image content. And of course, what you want from those images is several things, and I will focus on mostly 2 and 3 main topics. The first one is image restoration. That means really changing like the low-level image processing aspect of things. The second will be cell segmentation, so getting really kind of cell identities from those images. And the last one will be computational microscopy, which means, in a sense, just augment the process of actually acquisition with detection and inference pipelines as you acquire the images. And to start with the first part, Loic already gave you a rough overview of what it entails, and of course you're all familiar with that image, that you cannot, or that imaging itself has a lot of constraints, a lot of trade-offs that are mostly imparted by the sample health that you want to ensure. So a certain number of lights that you can impart in the sample, a certain speed with which you can acquire your images, and of course the spatial resolution that is given. So there's always a trade-off, and that leads to images that are not perfect. And that's just an example of it, of a planarian time-lapse. And here the challenge is that the worm, if you increase the light power to make the cells really visible, the worm just says, I don't want to be imaged, and crawls away, basically. And what we then did was just, okay, let's try just a stupid, very naive, supervised denoising approach. So we went to the microscope, acquired corresponding pairs of images in 3D of the same scene, and then just trained a neural network to restore those images, those high SNR images from low SNR images, which we termed then content-aware image restoration. And that worked quite well. So in this case, it cleaned up those images quite well. And obviously, at the final denoised images, cell segmentation is almost, well, it's not a trivial task, but it's way easier than in the original, where it was just impossible. And that was the main use case of this method, to create images that are then easier to analyze further downstream with any classical method. Of course, this, due to GPUs and everything running in like TensorFlow, PyTorch frameworks, everything benefits from GPU acceleration in itself intrinsically, so you can denoise 3D stacks quite fast. So everything scales quite beautifully. And of course, you always want to validate your images, and this is just showing that those nuclei that get restored are actually there. And you already see that there's a nonlinear transform of the intensities, as you see that the vignetting actually gets fixed, which is of course nothing that you want if you want to do quantitative photon count. So that's not something that you want to use for that. That's just an example that shows you that it's not only confined to denoising, of course, so you can do as well denoising and upsampling. So this is just images of a pupil imaginal disk that has a limited Z resolution. So here the sampling and the optical resolution both added to a roughly 6- or 7-fold decrease in resolution in the axial dimension, which was not good enough to delineate those membranes. And you can do with a similar approach by going to the microscope, acquiring images in one high and low SNR condition with a super sampling as effectively in the high SNR condition case, and then going back, training network, and going back to the live time lapses. You can actually recover that and get a beautiful delineation of those membranes, and it's then very easy to actually get the shape of that over time, or at least it was for the collaborators. Of course, the whole thing, and Loic already mentioned that, like the elephant in the room is always like that your machine learning pipeline is always as good as your training data, first. And secondly, if you deviate from, at inference time from your training data, which can be hard to detect, bad things will happen, right? So like implicitly in all the supervised methods, it's always that your training and your test modality comes from the same distribution, which means the same organisms, same structures, similar staining, same SNR, and so forth. And just to, and therefore you always need to be sure to validate your results properly by testing it or predicting on out of domain, or test not, or on stacks not seen during training. And just to showcase you what happens if you don't do that. So this is just an artificially really bad pipeline that we artificially created, where we denoised or trained on nuclei data that was shown like this. So this was the input, this was the ground truth, we trained the network, and we did the same for a membrane stain data set, and then we just got a nuclei model that is working pretty well on the nuclei data, and a similar membrane model that works pretty well on the flywing data. And now if you cross-apply them, of course, horrible things will happen, right? So the flywing model actually has never seen nuclei, and the nuclei model has never seen membrane. So that happens, right? And you don't want that to happen, of course. So with great powers come great responsibility. Loic already mentioned, like there are recently in the last 2 years or so, like a lot of methods popping up and were created by Loic, by Florian and other groups that are trying to get around that by not using a purely supervised approach, which always has this drawback that you're bound to your supervision, which means you're bound to your training data set. In a sense, to create denoising without high SNR ground truth. And the basis, or the first thing that started it, was a paper called Noise to Noise. Maybe already you know it, which means you just have the input are just two instantiation of a noisy image, and you try to predict one from the other. And actually, statistically, the best the network can do is predict the mean of this distribution. So you actually have to, you end up with a clean output, which is kind of interesting and was back then quite astonishing to see. And that gave rise to several methods that, for instance, noise to void or noise to self, that do that in a similar vein, even without two images as an input, but just within a single image as an input, by creating like a blind spot where you have to predict the single pixel intensity just given the neighborhood of an image. And that forces you to kind of remove, or because you cannot predict pixel independent noise, because that's statistically uncorrelated, or at least that's the assumption, you have to predict the mean of that distribution, which in the sense is your clean image. And that works quite nice. You don't have to have ground truth. It's still a bit worse than a supervised method, but it has the advantage that like this out of domain effect effectively is removed because you're training on the same images that you see. So the hallucinations are actually less pronounced, and you need pixel independent noise, which might not be the case, right? But there are methods out there that try to tackle that. If you're interested in these kind of approaches, everything that we do is open source, so go to this website. Or if you have a question, go to the forum. And with that, I come to the second part, which is cell segmentation. And as a computer vision problem, there's actually two different ways you can speak about segmentation in the context of cell segmentation. The first one is just finding out the foreground and the background of the object, which in computer vision terms is called dense segmentation. And what you normally actually want to do is instant segmentation, which is actually assigning every cell a different identity. And that's called instant segmentation, as I said, and that's typically what we want. And the challenges in microscopy are, of course, that you have often low SNR images. You have many crowded touching textureless objects, so that's a bit different to computer vision or natural images, where the opposite is most often the case. You have out of focus blur, which of course doesn't happen in natural images, or not that much. And these are all challenges that you have to address. So there are essentially two main lines in the computer vision literature to do this. The first one is bottom up. You first segment and then you localize. So like the original UNet was an instance of that, but as well CellPose, which is a great project, you all should check it out. It has a similar approach that you first try to find pixel identities. For CellPose, it's actually the flow vector. But you do something local. In this, for instance, in the UNet, you just want to figure out what is foreground, background, and then you do a clustering later. So you first do something local to the pixels, and then you try to group it into different instances. So that's the second step. And the other way around is top-down, so that's the other way around, effectively. And that's something that is very prominently present in the computer vision literature, that you first try to localize your objects with a bounding box, and then you try to refine it. So that's the Dectron, mask RCNN, so like your classical computer vision pipeline, YOLO, all the variants, they actually work in this principle. So YOLO doesn't do even a mask refinement, so it's just about the first part. But they first try to figure out the different instances with a bounding box, and then they try to refine this bounding box with something more precise. And that's a cool application from White et al. And Egelin that show that you can actually run like a localization network in real time on cells, which is always super cool to see. But what is of course the problem with that, with both of those cases in crowded cells, that they tend to merge objects in the bottom-up approach, and they tend to miss objects that are crowded because bounding boxes are just poor representations of your cells. And we try to fix that by doing something that is a bit better than bounding box, but instead of using bounding boxes, using polygons. So super simple idea, but very effective. We call it Stardust, and we predict densely both the centeredness of objects and for every pixel the radial or the distances along a set of predefined radial distances. So that's all. It's a very simple idea. So instead of bounding boxes, you predict polygons, and then you don't have to do anything anymore except for filtering overlapping polygons, which is called non-maximum suppression. But after that, you're done. So there's no mask refinement. Of course, this only works for objects that can be represented with that, with these kind of radial star-convex distances, which are exactly those that are star-convex, so objects that are roundish, potato-shaped, but not objects that looking like a boomerang or like this island we're on. And, but you can do that end-to-end. You predict it, everything, all those radial distances, object probabilities are predicted, and at the end they are filtered out by a second step, that is this polygon selection step, non-maximum suppression step, and after that you are ending up with all the polygon candidates that describe your scene. So it's a very, very simple thing. So if you create a toy task, which is voluntarily hard or made hard to screw up bounding box based methods, so for instance you do like these kind of shapes that have almost identical bounding boxes, what you will see for the bottom-up approaches, they're all fine. So they're all green. Green means that's a true positive. Blue means it's a false negative. Apart from the top-down approaches, where actually in those cases where the bounding box is overlapping almost 100%, just one of them gets picked. So that's what you. So that's designed to break those methods, and Stardust is fine with that. And in these cases, that's real data on the speed challenge data. You see, it's pretty good specifically in those instances where the cells are very crowded, very densely, and out of focus. That's just exemplified by this. So this is an example that I always like from collaborators in Manchester. So this is these are stem cells in a petri dish that are quite dense, quite noisy. And if you train, I think on 5 crops or so, a Stardust model, it manages to segment those pretty well. The same applies for histopathology. So here, again, you want to segment those cells. And you can do that on the whole slide in roughly 10 minutes. So that's like I think this was 3 gigabyte slide. And it's comparably fast, so you don't have to wait for hours. Of course, the same principle works in 3D. Instead of 2D polygonal distances, you just create distances along that are distributed roughly uniformly on a sphere, for which we use a Fibonacci lattice. And then everything just becomes a little bit slower because you have to first train rays in 3D. So instead of just 16 or 32, that well-described polygon, you have to use 96 or something. And of course, the non-maximum suppression of polyhedra is way more involved in polygons because you have to do a lot of nasty tricks to even do it in a comparable or in somewhat acceptable time. So you cannot, there is not really like an efficient algorithm for the intersection of star convex polygons. So there is one for convex, but not for star convex. So we do some tricks by approximating first the objects with several convex, like the inner part, the kernel you can do with a convex, and the upper bound and the convex hull is convex. And only if it in between the intersection, then we actually do the hard and important and computationally quite intensive rasterization test at the end. But this is just an implementation detail. So it works quite well for noisy data. So this is warm data from the Kindmiller lab and a crustacean data set from collaborators in Lyon. And even in those cases, it works quite well. So what we recently added is like the ability to not only delineate those cells, but as well classify those cells. So if those cells have not only identities, but like there's a predefined set of different classes, in this case 2 classes. Here, this is just the texture of those objects is essentially the class it refers to, so just 2 classes. And what you do is you just attach on your feature extractor, so that's a classical approach. So the first part of this network is like a feature attractor on which you predict the object probabilities and the distances, and you just attach another head that predicts the classifications, and then you aggregate them over the instance sets that you get, so it's quite easy. And at the end you get not only instant segmentation, but as well a classification, as long as you provide those at the training stage. What we asked it is something that is very, very important for large images, because for large images you cannot, like if you do it naively, and just compute all the polygons candidates at every tile and just put them in a whole box and then do a pairwise comparison, this scales quite horribly. So you see it's, the runtime is on the order of 1,000 seconds, so that's not good. But of course you can do certain tricks. You can speed up the neighborhood search, right, with KD trees. You can do sparse predictions, so you pre-filter those candidates per tile, and only those that really survive are taken into account, which brings down the memory. And then you can actually create like meta tiles that you do a complete full prediction on, and then you stitch those instances together on the overlap region, which brings down the memory requirement to almost constant time. And that's all included in the package, so you can use that. It's behind the scenes, always present. Another thing that we recently did is doing an augmentation package that works in 2D, 3D, GPU accelerated, being agnostic to TensorFlow and PyTorch. And that's just a package that helps with the training, because during training you always need to augment your data. So flipping, elastic deformations, noise intensity shifts, things that are typically necessary for microscopy deviate a little bit from what typically is available for PyTorch, right? Because everything is RGB 2D based, and that's not necessarily what you want. So if you're interested, you can use that. There are a lot of microscopy-specific augmentations like missing planes or Gaussian blur and all these kind of things that you probably have in microscopy. And it works for 2D, 3D. Again, it's GPU-accelerated OpenCL, agnostic to the framework that you use. And what we have recently started is gathering some pre-trained models, so using publicly available training data, in this case the data science ball that we already had, this too, for histopathology images, but recently we did one for budding yeast, because there were some papers that actually did the hard work of annotating data. That's always the hardest part, so I'm always grateful to anyone who's doing that. And then you can just train the Stardust model and distribute it. And interestingly, what we observed, for instance, here in this case for the fluorescence data set, it's actually a data set that consists of 4 sub-data sets for 4 different conditions, so small cells, blurry cells, the DSB data set, or this data set, which contains a lot of large cells. And what is interesting is if you now train a model only on one data set and then try how well does it work on the others, is depicted in this in this row. So this is the training set and this is the test set applied to on. So the diagonal is essentially like it tells you it works pretty well on the data set it was trained on. So this is this diagonal. But if you look at it off diagonal, some of them are quite horrible, right? The worst is if you take the train on this and then predict on this, and it's understandable, right? The images are like, the cells have a totally different scale, and this scale is consistent per data set, and that totally trips off the model, right? So that's understandable. But if you put them all together, essentially everything can be fixed. So it tells you just the capacity of those networks is so large that that's not a problem. So the more the merrier, typically. The same happens for the budding yeast data set. Again, here there's some black squares telling you that certain data sets are quite different from another. But again, if you put them all together, this accuracy can be rescued, more or less. There's still a slight drop between this line and the diagonal, but it's not that much actually. This is an example from data that just collaborators sent me, and just tells you how it would work. And that's just a pre-trained network that has not seen these. I mean, from that just was data that we got independently of the training data set. And it works quite well. This is just a video that we got from publication from this Wood et al. Paper. Just throw it on. Of course, it's not a challenging image, but it just tells you it's pretty robust to new images. Even if you add some noise to it, I mean, as long as during the augmentation you add some noise, it's pretty robust to that. We have, for these pre-trained models, you can try them out in Fiji. So we have a little Fiji plugin that we made, where you can just apply that. And typically for training you better need a GPU, but for inference a CPU is fine, fair enough. And as Loic already mentioned, we made a little Stardust plugin, and by we I mean mostly Uwe, so which is really something super nice, because the library anyhow is in Python, and Nepari allows you to really build very easily an interface to your existing Python library. So it's really a game changer. It's really cool. And time-lapse prediction. So this is a time-lapse of a budding yeast data set. And it works in the same way. If you just signify one of those axes as being the time, it will predict over time. It will try to match instances via a simple IOU matcher. So they should be more or less consistent if they overlap enough. And you get like a basic segmentation tracking without too much of a hassle within Nepari. The same works for 3D. just that you have to most likely train your own model, because it's pretty hard to do a 3D pre-trained model that works on all the nuclei and all the different asymmetries that you can have in lateral and axial dimension, which we figured out. So you probably have to train your own. And for the application or for the inference part, you better need a GPU again. And yeah, hit run. And then at some point, there should be, yeah, some nuclei. And thanks to Nepari, everything is rendered beautifully. So it was quite a pain before that. So thanks to Nepari team. If you're interested in that, again, we have a GitHub repo, and in the forum, you just use the tag Stardust, and then hopefully you will be helped if you have a problem. Finally, I just want to mention a short application of the whole machine learning-based approach to segmentation, which I quite liked. That was with Andreas Müller and Harald Tess and Janilia. And here the question was, they wanted to find out the organelle distribution in pancreas islet beta cells. So these are the cells in mouse that produce the insulin. And insulin is actually produced in vesicles that are quite spherical and quite abundant in those cells, and they are most likely transported to microtubules, or nobody really knows. So the aim was to segment all those organelles in several of those cells and see if there is a spatial correlation with the different organelles in between. So what Andy did, he actually sent some stacks to Janilia, and they acquired 6 or 7 cells in total of 2 different conditions with FIPSAM, 4 nanometer isotropic resolution. So this is the raw stack, how it looks like. So you can appreciate that this is a single cell, right? These are the vesicles containing the insulin. These are mitochondria. There's some Golgi appearing here and there here. So it's quite fascinating to see even that in the raw data in all its detail and all its glory. And there's, I always miss the centrioles. So these little small dots are ribosomes. And I always miss the centrioles. That's the nucleus, so quite nice. And of course, as you can already see, like the different organelles, they pose different challenges. So some of them might be easy to segment, others might be very hard to segment. And that's what we figured out essentially during this project. So naively we thought, okay, we just do everything with a unit, but then some organelles were easier than others. So for instance, nuclear centrioles we could do almost semi-manually. The mitochondria we could do with iLastic, which is great. And if it works, always use that. With a random forest as a backbone or as a backend. For the Golgi, we had to use a unit, so that was the only approach that really worked. The microtubules were really our biggest enemy, because that was not possible. So some poor person, Andy, had to manually label them all, which was just crazy work. But for the insulin secretory granules, so the spherical objects like Stardust 3D was a perfect match, and it actually helped a lot, because before predicting only like the foreground background and doing a watershed completely failed. And merged a lot of those cells, and this is just the prediction on those insulin secretory granules. And you see, you get nice instances that don't merge, which in this case you really never want. And of course, this allows you then to do a statistical model of all those organelle interactions, the lengths, the sizes, the interaction between the two. So you see that the microtubule length, that it changes with glucose conditions, that secretory granules are enriched near the membrane, and that the diameter of those vesicles is conserved. So all these interesting properties that were so far only visible by observations, but not by quantifications. And of course, because the output of Stardust is already a mesh, so you don't have to do even some marching cubes or so you can directly import it into Blender, for instance, and then give it to a talented Blender people that can work with Blender very well. For instance, Deborah Schmidt, so she's amazing, and she just rendered everything in Blender, overlaid it with the raw image, and I always like that image or that video sequence because it just shows you how tightly everything is coupled in between. So this, everything that is red are the secretory granules. The blue one are the mitochondria, just flying through. So that's all the insulin, so there's 10,000 of them per cell. That's the Golgi. The microtubule network, centrioles, nucleus, and so forth. And that you can do then for 7 cells, all the cells that you could, in a reasonable amount of time, image with a FIBSAM, because that takes actually longer than all the prediction and everything else, except for the microtubule annotation. And that you can do for 7 cells, which I always liked and find quite amazing that you can do that. Finally, I want to quickly mention 2 little projects in the realm of computational microscopy. So using machine learning to get better aberration estimations, or get faster way to estimate aberrations from PSF stacks, and something that colleagues at the EPFL are currently doing, which is called event-driven smart microscopy. So the first project is a project with Debaya and Sahan from Dresden. And he asked, first he observed that the fact that probably everybody knows that in whole organelle imaging, the PSF is actually changing throughout the whole tissue due to aberrations that are sample specific. So typically if you embed little fluorescent protein aggregates that serve as point sources and just image that, you see that the point split function actually close to the camera is way better than it is behind the camera, so that's probably a fact everybody knows. So when you want to, in any way, to do like any aberration correction, be it in hardware or be it in software, you of course have to know this aberration, and you have to know it on several points, on potentially 1,000 points in your sample. And the question is how to do that efficiently, such that they can actually do that. So 1,000, on the order of 1,000 aberration estimations. And that was the question Debaya asked. So given like a bead-like image, you want to estimate the aberration, in this case the wavefront aberration, and for extracting aberrations from 1,000+ locations of bead-like structures. And there are classical PSF modeling packages that does that, for instance Zola 3D, which is amazing. It works really, really well. The only bottleneck is, or the only problem is that it's a bit slow if you want to do it for 1,000 or 10,000, so it's not really a very fast way to do that. So the question was, can we create a fast and accurate aberration prediction model to do that on the GPU without the need of experimental training data? Because if you don't, of course, want to go to the microscope, like do a lot of PSF measurements and actually do a Schack-Hartmann sensor and do all that, because you're never going to make it to 1,000 images of that sort. And what Debaya then thought of, okay, and that's an approach that has been done by 2 or 3 other projects in the last 2 years, similar to ours, is that instead of using training data that require a microscope, you just create a very good PSF simulator, and you just create your own training data. And that's what Deb was doing. So the training was done on synthetic data, the output was directly regressing on the Zernike coefficients, and then you apply that on the real microscopy data. And that actually worked quite well. So if you have the ground truth waveforms here, for instance, we did some experiments with actual Schack-Hartmann sensor and provided data where we had the ground truth of bead-like structures, and you compared it to Gershberg-Saxon or Zola, like what you get with what we call PhaseNet is pretty much similar to what you get with Zola. It's a bit worse, but it's on the order of like roughly 20 nanometer of deviation, which is acceptable, or we deemed it acceptable. But the runtime to acquire that or to infer that aberrations, of course, is orders of magnitude faster. So you can actually do that in a batched way for 1,000. So this is just the inferred aberrations for single mode aberrations, when you really kind of introduce a single aberration, in this case astigmatism, and you just change that from a large to a positive value with no aberration here, and just ask what is the predicted and what is the experimentally introduced aberration. In a perfect world, this is a line, and we see there it's pretty much watching well, except for large experimentally introduced aberrations. Of course, you can ask how many of those C-planes you actually have to provide, and that was just an interesting ablation study. So depending on the symmetry of your aberrations, which is understandably, depending on those symmetry, one plane is enough or one plane is not enough. So for all the even, one plane is not enough, which is clear because it's effectively, for instance, defocus is symmetric, right? So you don't know if it's defocused or in front of you or in the back of you. But for all the others, you can get away with a very, very low amount of frames, actually. And the second part is a project that was done by Suliana's lab, and I had the opportunity to work with them a bit on that, and driven by Dora and Willy and Juliette, who's in the audience too. And that's about like doing a smarter way to image mitochondrial fission events. So they are looking like this. So typically you have a field of view, for instance, with an ISIM. In this case, they used an ISIM a lot. And you image mitochondria. in a cell, and you're interested in constriction events. These are events where the mitochondria are about to divide, and sometimes they don't divide. They just get confined. They look about to be dividing, but then they don't divide, and sometimes they divide. And these are the biological interesting events that this group wanted to find out. So here, for instance, that's an event where this mitochondria is very faint. I mean, you can see it barely here, but it's almost about to divide. So it could be that's a constriction that could go into the fission mode or into no fission mode. And these are the interesting, biological interesting events they wanted to observe. The question is, if you image slowly, then these constrictions dynamics, and slowly means like with 0.2 hertz in this case, the constriction dynamics are very roughly captured. So if you want to measure, for instance, this diameter over the course of time and just fit what is the smallest diameter, you might miss the smallest diameter just because of your temporal sampling. But the sample or phototoxicity and photobleaching is quite well. If you now image fast, that would allow you to acquire the constriction dynamics perfectly. Then you see a horrible photobleaching. So that was actually the obvious constraints they wanted to solve. And the idea was quite simple, that you adapt your temporal sampling essentially to the events that you observe. So you only use fast events, or you only image fast in those cases, in those rare cases when there is actually a constriction event happening. And for that, you actually have to know that, right? So there are some perception tests involved that has to be carried out fast enough to be involved with the actual acquisition process. And what they did is essentially they measured not only the mito signal, but as well a second channel called DRP, which is required for fission but not sufficient. So it pops up whenever there is a fission, but sometimes pops up somewhere else. So that's kind of a helper to do the annotations, because just clicking on images would be horribly inefficient, because it's just, you don't want to do that to a human. So we helped ourselves a lot by using this DRP as a guiding channel. So this DRP signal was guiding or giving potential candidates of fission events that were then manually curated, and from that we generated like event probability maps that really indicate what are constriction site events that we want to predict given these two images. Then we just train a network and then put it on a live system, and as you see here, it works quite well by spitting out like those potential constriction events. And whenever the probability map that was spit out or that the network predicted was above a certain level, we deemed there to be an interesting event in the field of view, and then the temporal acquisition sampling of the device was just increased to 4 frames per second. So it's quite a simple idea. So if you go here in time, you see that here, at certain portions of times when there was a constriction event detected, the temporal sampling was increased to roughly 4 hertz. And when there was no event anymore detected, it went back to the slow mode. And there was saving a lot of photons, and photo bleaching was fine. And that's a time-lapse. And here you will see at the top left corner, you will see a little symbol if we're in the fast mode, and there will be no symbol when you're in the slow mode. So that's the slow acquisition mode. So every 5 seconds there will be a frame. And now here pops up a little constriction that you see here. It goes into the fast mode, acquires with 4 hertz the constriction event, and then if the inferred probability goes back, it goes back into the slow mode. And that of course allows you to capture way longer the dynamics of something where you don't have to be in the fast mode all the time. Of course, this allows you to really capture, compared to the slow mode, to really capture this minimal constriction site, as I mentioned, like with a way better temporal frequency, thereby not missing the smallest constriction site. So you get a better and a smaller constriction site minimal width. And at the same time, the bleaching decay or the cumulative light dose that you get with it is vastly decreased. So you have actually in this matrix that I showed you, have 2 green checkboxes there, which is quite nice. And with that, I want to thank everyone that helped in those projects. I want to thank the EPFL, and I'm happy to take your questions.
Okay, thank you, Martin, for this nice talk. Now it's time for questions.
Martin, thanks for this great talk. So it is quite amazing to see this segmentation of all these nuclei in these developing embryos. But people working on morphogenesis, they're often interested in cell shapes, so they're more looking at membranes more than nuclei. And these cells are very much packed. Eventually, they adhere one another. So, did you have the chance to tackle this issue? Because we're then interested in, defining apical size of cells, basal, topological changes of cells, those kind of things.
So, for non-convex, essentially objects, right?
Yeah, for entire embryos, in fact. So for
segmenting a single embryo,
segmenting cells within embryos, not nuclei but cells.
Okay. I mean that's something that so essentially that's an interesting question in a sense, because what we predict is essentially the affinities to be at the border. So it doesn't tell you necessarily that you have to take a single pixel and just draw the polygon, but you can actually convert whatever you get as the distances into kind of an affinity-based map. And if you, for instance, watershed that and do some pre-processing on that, you can get actually what you were asking for. Already out now of what Stardust would give you is just like the process that you would have to take is a bit more problem specific, such that you cannot do it like in a general purpose way. But we actually did that in a recent preprint, which will soon be out in terms of expansion microscopy cells that where really those cells were not convex, and we essentially used the affinities predicted from those star distances to segment non-convex objects. And you have already access to it, but you have to dig into the code essentially to get that. So it's a bit more involved. So that's currently more like a hacking topic.
Thanks.
Yeah.
Hi, great talk. I have maybe a naive question, but from what I know about CNN, which I use a lot in all these presentations this morning, I think CNN considers that essentially the data has the same resolution in X here in 3D, for example, X, Y, and Z. And so how do you deal with, if you use a 3D unit, for example, how do you deal with the fact that in general, images don't have the same resolution in the Z direction?
So that's actually, so in XY, for instance, you can get away with that because typically not X and Y, like they're typically isotropically scaled, but essentially for 3D, for most of the 3D segmentation and not only Stardust, but as well just the UNet approaches, it's really hard to do something that is generalizable to any anisotropy. What people typically do is they rescale their network to a common anisotropy, which 1:4 or something, which is kind of reasonable for normal purposes. But in generally, that's one of the crucial bits that have to be tackled to do that, right? So either you rescale to something similar, or you have different networks for different resolutions. So that's essentially it. But it's exactly what you say. It's a problem for 3D. Yeah.
No more questions? Okay. So thank you again, Martin, for this nice presentation.
Don't run away, please. Don't run away. we will have a presentation by Edouard Bertrand now on France Bioimaging, and I think David is also telling us something about the GDR ISIS, if I remember well. Okay, so. Yeah, and I would take the opportunity now to thank Alan also. Thank you very much for chairing the session. It's really a great thing that we have young scientists who help us and who chair the session. I think it's a great experience for you, but it's also a great help for everybody here. Thank you.
Okay, thank you. So I will give you a short presentation about France Bioimaging infrastructure. So first I would like to thank the organizer on the GDR MIFOBIO to giving us an opportunity to present some of the things that we are doing. So I have to say also I'm very happy to be here. I love MIFOBIO. I think it's a great experience for everyone. So you can listen to all this great technology and actually practice them, look at the setup and etc. And I think the difficulty is actually when the meeting is over and you go back home and you want to practice some of this technology. You know, sometimes if you are lucky, you can access to them fairly easily, but sometimes if you are less lucky, you cannot access them so easily. And that's where I think the infrastructure comes into play. So we're a national infrastructure for biological imaging. So our mission is to provide access to imaging technology to the entire national community. So everyone, of course, we want to serve everyone wherever you are. And so our motto is innovation, access, training. So innovation because we want to provide you access to the most developed and most advanced technologies. Training, of course, on access because then you can come to use our instrument on technologies and acquire data that are useful for you. So we have both R&D activity and infrastructure activities. So in a nutshell, France Bioimaging, so we have about 20 core imaging facility and about 50 research and development teams. So the whole idea of the infrastructure is to associate research and development team with core facilities such that the new development can be made accessible to the entire community through the facilities. So we are a large infrastructure, so of course we have a lot of people, lots of setup and a lot of users. And the way we are structured, so we have nodes, what we call nodes. We have 6 geographical nodes, Marseille, Bordeaux, Montpellier, Nantes, Rennes, and Paris area. On one transversal node, which is a bioimage informatics, especially important for this module. And one thing I want to mention is that the perimeter of the infrastructure is not fixed, it's evolving. So we had a couple years ago a call for a new node, and in 2020 we welcomed the Bretagne Loire node, which reinforced the infrastructure on several important aspects. So another important thing is that France Bioimaging is the French node for the Euro Bioimaging, which is a European research infrastructure, and we are also part of Global Bioimaging. Okay, so I want to say a couple of things about action that we are implementing now. So first, about access, our main goal is to improve the access to the infrastructure. So for this, we started to subsidize access for the external user that want to come to our facilities to do experiments. So we provide 750 € per week that cover instrument usage. On the way to access it is fairly easily. You have to go to the Eurobioimaging portal and fill a form. And in fact, if your project is already financed by an INR or whatever, you go on a fast track, and then it's always accepted, and you get in contact with the facility where you can set up, the practical stuff to come and do your experiments. So 2 other things about access. So first I want to mention that currently Eurobioimaging has a call for transnational access. So if you want to do experiments in a different country because some technology is not available in France, so you can apply to it. So you will have 5,000 €, and the deadline is in December 15. And one last thing about access also is that because we are part of Eurobioimaging, so there are specific calls, European calls for European research infrastructures. So we will benefit in the future from money to cover access to France Bioimaging. So this is currently, we have already within Eurobioimaging applied to a couple of calls. I have to say that it will be a small amount of money on specific teams, but we hope that in the future, if the concept, turns out to be successful, we'll have more money to cover a lot more access to the infrastructures. Okay, about innovation, so we have several innovation axes, so in particular on data, which I will develop in a minute. So what we do is some scientific animation. So what I want to mention is that we are starting some new technological working groups, and we chose to target areas that are not covered by other existing structures of the GDR or the research network. So one, on light chip macroscopy started in 2019, is very active, and in early 2022, we will start a working group on intelligent imaging, high content multiplexing, and also small animal and clinical macroscopy. And I also want to mention one initiative at the frontier of France Bioimaging on Frisbee, on electron microscopy, where the topic will be in situ structural biology, and that will start early in 2022. Okay, what also we do is we take action to stimulate tech transfer. So we have a person in charge, which is Etienne Henry. So last year we had an internal call that we call a tech transfer. So the call was specifically targeted to have transferred technologies from the R&D team to core facilities. So we targeted mature technologies. So we financed 5 projects. So I will not go into the details. So one, very briefly, one was an instrumental project for a high content single molecule screening platform in Bordeaux. On the other 4 projects were in fact about data analysis and were general tools. So one was with IC to implement some deep learning component on new segmentation algorithm. One interesting one also is called Morphonet, is to develop an interactive morphological browser to explore complex multiscale data. On one is called, that was is by Emmanuel Faure. On one on Bioimage IT by Charles Crevran is to develop an open source interoperable framework to integrate image data management to analysis using advanced IT solution. So we chose this project because we think that they are of high value for the community and everyone will benefit from this development. I also want to mention that currently we have another call ongoing. It's called Outreaching because it's targeted towards the external user. So the idea here is to target immature technology, so that need to be developed. And what we'd like to have is a pair of applicants, one external user that has a specific need that needs to be addressed with the new technology, and then a research team or a facility from the infrastructure that will team up with the external user to address this technological challenge. And the idea will be to finance 2 or 3 projects with about 80 kilo euro for each project. And you can, of course, contact us if you want more information. The deadline will be the end of April. So we chose a long deadline because we think that you need time to actually find your external user or the team that will be willing to develop some technology for you. Okay, and finally, one thing that we're doing is to implement new resources. On this, we have an important plan in data management, analysis, and visualization. So we want to take 3 actions. The first one is on data management. So it is an action that is driven by Perrine Polgilotto and Emmanuel Faure. And the idea is to develop the data verification, access to high-performance computing, and develop the use of Mesocenter for storage and computing. So we'll have early in 2022 3 positions that will start on temporary contract, but we should be able to have a stable position after that. 2 will be in Nantes, 1 in Montpellier. We expect 2 more positions in Paris a little bit later. And one important thing is that these people will work within the infrastructure, but for the entire community. So the second action, which is a very ambitious plan, is to develop a remote image analysis service. So this is driven by Jean-Yves Tinvez and Anne-Sophie Mass. So this is very ambitious, but we have some force and some strength to rely on. So first, we can rely on the image analysis of hub of Pasteur, which is very well structured and function already very well. And we also have other resources within the infrastructures. And the third action is tool development, of course. So the main direction is to develop artificial intelligence and deep learning tools. And the other direction is to develop tools for visualization of complex data set. So a lot of it will be done by, of course, research teams. But I want to highlight 2 positions that we will have, 2 engineer positions. So the first one will work on developing annotated image database. We heard earlier in the talk that is a limiting thing, for people developing deep learning tools. So we will have a person to do that, and that will be associated to R&D challenges that will use this database. And the idea is to mobilize the entire community to try to find the best tool to address a specific technological question. So for this, we'll have a mobility at the CNRS Open very soon. And the second position will be complementary to this one. And in fact, it's another engineer that will be in charge to implement the best tools that will be selected towards these challenges in popular software platforms such as Fiji or IC or others, such that these tools can be used can be accessed in a very easy and user-friendly manner for biologists and people that are not versed in scripting and things like that. Okay, so our 4 key actions for the next years will be to improve access, stimulate R&D, and implement new tools for data management, analysis, and visualization, and also some training action which I will now develop. And to conclude, I just want to insist that France Bioimaging is a resource for the entire community. The second thing is the infrastructure is evolving, and you can actually shape it by participating to its action and by giving us your feedback. And you are welcome, of course, to contact us at any time. And I thank you for your attention. Stay here. There will be another short presentation.
J'en profite juste pour dire une paire de lunettes. Merci. Dans la salle, à côté de la salle de culture. Une paire de lunettes dans un étui noir.
Ok. Juste un slide très rapide avant de clore la session. On est ici donc avec le GDR MIFOBIO, qui est un GDR très interdisciplinaire sur les aspects imagerie pour la biologie. Donc, il existe plein d'autres GDR et un des plus proches, possiblement très intéressant pour la communauté ici, est le GDR ISIS, Images, Signaux et Vision, qui existe depuis de très nombreuses années, qui a des thèmes avec de nombreux points communs avec notre communauté, sur de la modélisation physique de l'imagerie, sur de l'imagerie computationnelle, sur des problèmes inverses, de l'apprentissage machine, traitement d'images. C'est 194 labos en France. C'est les sections CNU, du traitement du signal et de l'informatique. Je pense que ça peut être très utile pour vous quand vous cherchez des étudiants, recrutement de thèse, postdoc, du côté plutôt de ces thématiques-là, où il y a un peu moins de biologie et beaucoup. Donc, pensez à aussi diffuser vos offres via ces GDR pour permettre une intercommunication entre nos nombreux réseaux. Et alors, je mentionne aussi, puisqu'on a une dizaine de ce GDR présent là, qu'il y a une journée particulièrement importante et qui a un overlap fort avec notre communauté ici, c'est la journée des imageries optiques non conventionnelles, JIONC, qui existe depuis 2004. Et alors, c'est particulièrement intéressant pour tester des technologies matures d'un point de vue de l'instrumentation, mais qui n'ont peut-être pas encore été testées de manière très approfondie sur des échantillons biologiques de choix. Des fois, les gens tapent dans du jambon, etc. Donc, je suis sûr qu'il y a des mariages très intéressants à faire. Et Corinne Fournier et Julien Fad, qui sont dans les organisateurs, sont ici avec nous cette semaine. Donc, n'hésitez pas à venir vers nous pour faire évoluer ces journées en incluant plus de couplage avec MIFOBIO. Voilà, merci. Et puis donc, on remercie tous les orateurs de la présentation et avant d'aller déjeuner. Au revoir.